All right, hi. So I'm Alex. I'll tell you about some of the work we're doing in the lab. So our work is on multiplex antiviral drug discovery. Great. So I'll give you a quick overview of the talk. I'm going to talk about the background on drug discovery, our approach to drug discovery, quick snapshot of our data, and then some future directions. So a little bit about drug discovery. So typically when you do drug discovery, you do it against a single cell line or a single protein at a time. And that's good, and you spend a lot of time and money and effort, but you really only get one answer. And that's a little bit of a problem for coronaviruses because there's hundreds circulating within animals, many of which may have the potential to jump into humans. And so we think it's great if you can find an answer, obviously, to address the current problem, which is SARS-CoV-2, and we're obviously very interested in doing that. But this has been the third coronavirus in about 2 decades, and so we really think it's only a matter of time until another one comes out. And so what our goal is, we're really interested in trying to identify some sort of broad-acting drug against many coronaviruses. And so what are we looking to target within coronaviruses? For our group, we're particularly interested in the Corona 3CL protease. It's within all coronaviruses, they all have this protein, and it's required for the virus to kind of do everything it needs to do to be a virus. And in addition, the idea of drugging proteases is not new. There have been quite a few successful stories of protease inhibitors for both HIV and Hep C, which have really transformed the way we treat those patients. And so our goal is to identify a broad 3CL protease inhibitor. And to achieve that goal, we basically kind of realized we needed to change the way that drug screens are performed. And so we needed to develop some sort of method that let us look at not just a single protease, but dozens of proteases at a time. And so how do we do that? How do we do our screens? We use a very simple model, which is yeast as our screening chassis. And it turns out if you take yeast and you put a viral protease inside them, they become very unhappy and don't grow. If you take those yeast and you grow them in the presence of a protease inhibitor, they become happy and they grow very well. And so this lets us use yeast as sort of a biosensor of protease activity. The yeast is not growing, protease is active. The yeast is growing, protease is inactive. And so this lets us generate dozens of different protease expressing yeast strains, each of them with a different protease that we can study. Now the problem is, how do we study them all at the same time? And so to do that, we use DNA barcoding technology. And so the way that works is I take a bunch of different little yeast strains, and I give them each a unique DNA sequence that I know exactly what that is. And so we insert that into their genome. And we know exactly where we're inserting in the genome, and we know exactly, again, what the sequence is. Going forward in this talk, every time you see a different colored yeast, it just represents a yeast cell that has a unique DNA barcode. So now what I can do is I can take the little blue yeast, and I can give them SARS-CoV-2 protease. I can take the teal yeast, and I can give them MERS protease. I can take the black yeast, and I can give them SARS protease. And I can take the yellow yeast, and I can give them a control protein, in this case, yellow fluorescent protein, which when expressed doesn't make the yeast unhappy at all. If I take a mixture of these cells that are equally represented 1 to 1 to 1 to 1, and I don't express any of the proteases, the cells grow just fine and they maintain that 1 to 1 to 1 distribution. And I can see that by simply sequencing out DNA barcodes within the population. If I then take my starting pool again, this time induce protease expression, what I see is that all the guys that express proteases, so the teal guys, the blue guys, and the black guys, they don't grow as well, but the yellow guys grow really well because they don't express anything toxic. And again, I can see that all very easily using DNA sequencing. Finally, if I take a starting pool and I induce this expression, but this time I grow the cells in the presence of a known SARS protease inhibitor, what I see is that the yellow cells and the black cells increase in their abundance. And again, I can see that very easily using DNA sequencing. So to cut to the chase, I'll share with you guys some pilot study that we've done. In this pilot study, there's 50 barcoded strains tested against 96 compounds, which is about 4,800 tests are being performed. And we fit this all into a single 96-well plate. And so I'm only going to zoom in onto a very small widget of that data. And so on the x-axis, I'm showing you a variety of different viral proteases. On the y-axis, I'm showing you a bunch of different drugs we tested. And then in different colors, I'm showing you how confident we are in the drug actually affecting the protease. The darker the color is, the more confident we are in the call. And so this is what the data look like. And if we just highlight a few aspects of it, this column here represents HIV protease, and every single black square represents a known HIV protease inhibitor we're able to detect clearly working and stopping the protease activity. In addition, we had a known rhinovirus protease inhibitor, and we were able to also see its activity against rhinovirus protease. So what are the next steps for us? Well, we plan to kind of increase the scale of our assay. We're going to hopefully develop a library of about 70 viral proteases. I think we basically already have that, of which about 20 are coronavirus proteases. And we're basically gearing up now as a group to do a greater than 10,000 compound screen. In addition, we've been focusing on this coronavirus 3CL protease, but it turns out coronaviruses have another protease called the PLP protease, And we're working with Rodney Rothstein here at Columbia to see if we can develop an assay to actually be able to screen for inhibitors against that also. With that, I'd like to thank a few people. So obviously, I'm not an island unto myself. I have really amazing people I work with, and those are the students that lead this project. In particular, Debbie Hong, Shoei Katani, and Sam Resnick have really been critical in driving this project forward, since without them, there is nothing. With that, I'll take any questions.
Thank you, Alex. Let me see if there are any. I think there are a couple of questions.
Alex, it's Sagi. Just a quick question. I just had a thought. So are you actually looking at just human infecting viruses or also bat coronaviruses?
Yeah, so there's only 7 human coronaviruses that I'm aware of. So actually within our library, we have a variety of other ones. Some bat, we have beluga whale. We just have quite a variety, and we're actually gonna try to enrich that even more.
Yeah, the reason I'm asking is because, you can imagine perhaps moving this into human clinical setting might be too far out, but in terms of prevention of new outbreaks, you can imagine giving it to bats or spraying areas where bats eat and stuff.
Yeah, I agree. You can definitely imagine that. I think the other thing we're thinking is that obviously the next coronavirus is probably going to come from animals, and so we might as well have a compound that we know straight up works on that, because no one's really has the throughput or the assay to be able to do that, and we think we do. But I agree, maybe you can also figure out some delivery method to actually be able to sanitize the population. It's possible.
Awesome.
Alex, how do you deal with the specificity of the compound? So right now you have a variety of proteases from different viruses. You also have proteases from mammalian species as the sort of negative controls?
So actually, we've thought a lot about this, whether or not you put human proteases in it. And right now, we don't have any human proteases in the assay.
So what do you use as negative control? You just look at the specificity within your set?
Yeah, so we look at the specificity within the set of viral proteases we have. We also have a series of yeasts that are, I didn't talk about this, they're actually specially designed to sense off-target activity. So we have a series of yeasts that we've mutated to make them hypersensitive to perturbations in core cellular pathways. And so we think we can get some idea about whether or not this drug causes DNA damage, mitotic arrest, things like that.
Thank you.